we're live.
Great. Welcome back, everyone, to Elasmo Week. I'm your host for the week, Dr. David Schiffman. Elasmo Week is an online outreach event about the science of elasmobranchs, shark skates and rays, and the scientists who study them. This week is all about diversity, highlighting the diversity of scientists, the many ways of doing elasmobranch research, and showing off as many weird and wonderful species as possible. Elasmo Week wants to make a platform where real scientists can share their love and work with the public. It's my pleasure tonight to introduce to you Dr. Dean Grubbs, an Associate Director of Research at Florida State University's Coastal and Marine Laboratory. Dr. Grubbs is a fish ecologist with interests in the biology of exploited and poorly studied estuarine and marine taxa. Most of his research addresses specific biological gaps necessary for the conservation and management of coastal and deepwater sharks and rays. Dean's interest in sharks stems from being raised on North Florida's Gulf Coast. He received a bachelor's degree in marine science and biology from the University of Miami and a doctoral degree in fisheries science from the Virginia Institute of Marine Science at the College of William and Mary. Dean was a postdoctoral researcher and faculty member at the Hawaii Institute of Marine Biology before moving to Florida State University in 2007. He is currently a research professor and the Associate Director of Research at the FSU Marine Lab, as well as a member of the IUCN Shark Specialist Group, the NOAA Small-Toothed Sawfish Recovery Team, and NOAA's CDAR Advisory Panel for Highly Migratory Species. He's also a scientific advisor for the Save Our Seas Foundation and is the immediate past president of the American Elasmobranch Society, the world's largest scientific society dedicated to the study of sharks and their relatives. Dean's research has been featured in many television documentaries, including National Geographic TV, the Discovery Channel, and PBS. He's taught college courses on the biology of sharks and rays for more than 25 years, and with Dr. Dan Abel, he recently co-authored the book, Shark Biology and Conservation, Essentials for Educators, Students, and Enthusiasts, published by Johns Hopkins University Press. Take it away, Dean.
All right, thanks, David. I'm going to share my screen now. Hopefully you can see that. Okay, thanks for that introduction, David, and thank you to the Elasmo Week organizers and to Miss for putting this all together and for inviting me to give this presentation tonight. And thank you to anybody who decided to join on a Friday night. I appreciate it. So as David mentioned, I mostly study the biology and ecology of coastal and deep sea fishes, primarily sharks and rays. Most of the work we do in my lab is related to management and conservation. We do a lot of work on population dynamics, life history, strategies, habitat use patterns, a lot of tagging and telemetry. We also do a lot of work on community structure dynamics, as well as when we start talking about endangered species such as small-toothed sawfish, recovery and research, particularly to promote recovery of those species. If you tuned in, I think Sunday night to Jasmine Graham's presentation, she's a recent graduate with a master's degree from my program who is doing work on recovery of small-toothed sawfish. So the work we do in my lab is concentrated in three general areas. We do a lot of small-toothed sawfish recovery research, as I mentioned, in both the Bahamas and the United States and Florida. A lot of coastal shark research throughout the Gulf Coast of Florida primarily, some in the Atlantic coast. And then we also do a lot of deep sea ecology work, concentrated some in the Bahamas, but a lot in the Gulf of Mexico and a few other places around the world. And so tonight I'm going to mostly concentrate on some of the deep sea stuff and tell you a little bit about sharks of the twilight zone primarily. So sharks that live in the deep sea, some of the groups that don't really tend to get focused on very much on most of the documentaries you might see on television. So if you ask somebody to name a typical shark, they're almost certainly one of the first things out of their mouth is going to be this thing, this white shark, it's I don't even find it attractive. It's not even a very interesting shark to me at all. And if you ask them about how many species of sharks there are in the world, how many different living species, most of the general public doesn't really know. And if you ask them to start naming them, they're probably going to name some of these on this slide: white sharks, bull sharks, whale sharks, tiger sharks, hammerhead sharks, the things that most people are familiar with. Truth is, these are not typical sharks at all. And there are actually about 540 species of sharks in the world. And that's just sharks. That doesn't include their close relatives, of course, the skates and rays, or their slightly more distant relatives, the chimeras. But most people are unaware that, when you ask someone to describe a shark, they'll describe a big, toothy predator that lives toward the top of the food chain in the ocean. In reality, most sharks are not top predators, and most sharks don't get very large. The majority of shark species in the world get a maximum length of less than about 3 feet, less than about a meter. And about half of the living species of sharks live their entire lives in the deep sea, and we define that as deeper than 200 meters, deeper than about 700 feet deep. And here are a few examples. In fact, we did an analysis in 2005, Chip Cotton and I did, where we found that roughly half the species of all chondrichthyans, so skates, rays, sharks, and chimeras, live their whole lives deeper than 200 meters. But we know almost nothing about the basic ecology of nearly all of those species. Life history data are available for only about 5% of them. There's limited data on habitat use patterns and movement patterns for a few species. And one of the things we found is that only about 1% or less than 1% of all publications, scientific publications dedicated to sharks and rays are dedicated to the deep sea taxa. This graph basically shows the number of scientific publications through 2015 in gray, the total number on some sort of shark, ray, or chimera, and the black are the ones that are on the deep sea species. And you see it's a pretty small group that are concentrating on the deep ones. And so you might ask, well, why is that? If they make up half the diversity of sharks in the world, or chondrichthyans in the world, why is so little focus paid to them? Well, some of it stems back to some early hypotheses that proved to be completely wrong. For example, there's the Azoic Hypothesis that came out in 1859 and several other similar hypotheses around the same time that basically stated there was no life in the ocean deeper than 550 meters, about 300 fathoms. Said the deep sea is basically so dark and cold, the pressure so high, it's anoxic, low oxygen. So how could life possibly survive? And so that really drove biologists sort of away from trying to do anything in the deep sea. But of course, the Azoic Hypothesis was completely disproven by the HMS Challenger expeditions in the late 1800s. And they basically found that there were animals down at least to 5,500 meters deep. They published the findings of these expeditions in 50 volumes, almost 30,000 pages. But almost all of this expedition was dedicated to invertebrates, not to fishes, and particularly not to the sharks and their relatives. The other reason you don't see a lot of concentration on sharks and their relatives is it's not easy work. It's logistically hard to work on the deep sea animals, particularly the larger ones. This is a little cartoon from a National Geographic documentary that Toby Daly Engel and I did a few years ago. And we were doing a show on six-gill sharks, and they're basically drawing an analogy that what we were trying to do is fish from the top of the Empire State Building and try to catch sharks swimming on the street below. And of course, this ignores the fact that actually, that street had nothing on it. There are no other obstacles in your way. And you're sitting on a stable platform, not on a rolling ship in rough seas. And also, the height of the Empire State Building, that's about the shallowest of the deep sea sharks. Some of the work we do is 7 or 8 Empire State Buildings deep. So it's logistically challenging to do this work, which also means it's expensive. Fortunately, we were given an opportunity. It's unfortunate that the BP oil spill occurred in the Gulf of Mexico, but from that oil spill, we were able to get funding to do a lot of deep sea work in the Gulf of Mexico. This is my student, Joanna Imhoff, some of you may know. They're measuring a large bluntnose six-gill shark. And this allowed us to do a number of research cruises, 16 to date. We hope to do another one in the next few weeks, to look at the effects of the oil spill on deep sea sharks and bony fish. I'm not going to really talk about that work, but it does provide the mechanism to describe some of the things that we've found and let you see some of these deep sea critters. We basically came up with a method that's highly repeatable, that allows us to set a lot of different hooks and traps on the bottom for a very defined period of time at a very targeted depth. And we're able to repeatably catch both small and large bony fish, as well as sharks on these sets. And so to date, we've done 16 research cruises, over 500 long line and trap sets, basically from 186 to over 2,500 meters deep in the Gulf of Mexico. This shows you basically the you see Gulf of Mexico that's associated with the deep sea. Basically the temperature dropping from around 17 or so at 200 meters. By the time you're 1,000 meters, it's about 5 degrees C, and it's between 4 or 5 all the way down after you get below that. And so what we find, you know, as I mentioned, most of the basic ecology, for most species, basic ecology is unknown. So that's one of the major reasons to study the deep sea chondrichthyans. We need to know more about their ecology, their biology. And what we do know so far is they're extremely vulnerable to anthropogenic insults such as oil spills or overfishing. We also know that as coastal resources have become more and more fully exploited, if not overfished, and as technology has advanced, this has driven more and more fisheries to exploit deep water resources. On top of that, this is an article I wrote on mortality and associated with bycatch in fisheries around the world. You can download it for free right there, Save Our Seas magazine. Bycatch in deep sea trawl fisheries may be the biggest source of mortality for deep sea chondrichthyan fishes. For example, here's a super cool deep sea shark, Trigonognathus, the viper dogfish. It's only known from a few specimens until a couple of years ago. Some were caught in a trawl net, basically in Taiwan. Super cool fish. We don't know anything about this thing. So we're behind the curve in trying to figure out how to manage these species against insults such as bycatch overfishing. What we do know is that bycatch mortality is very high. This was a study that my student Brendan Tallwer and I did a few years ago, looking at 3 deep water species in the Bahamas and comparing their at-boat mortality, so how many of them are dead when you bring them to the boat, versus if you release them, how many of them die after they're released when they are seemingly fine when you release them. And you can see it's basically 50 to 88% of the sharks die from being captured and brought to the surface. And I'll get back to that in a little bit. The other thing we do know is that the deeper the sharks occur, typically the more vulnerable they are. On this graph, you've got the age at reproductive maturity on the x-axis, and basically their ability to rebound from being overfished on the y-axis. The black dots are deep sea species, the white dots are shallow species. You can see the black dots across the border pretty much below the line of the white dots, meaning they're more vulnerable to overfishing. And if you look at the maximum depth of something occurs, you can see that the shallower water species tend to rebound faster from overfishing than the deep sea species. There's one just really quick example from Brian Moe's work in our lab here at FSU on Centrophorus uyato, a gulper shark. He's aged them to at least 76 years. This is a species that reproduces, has at least a 24-month gestation period, and only produces a single pup. So imagine if it takes 2 to 3 decades to reach sexual maturity, and it's only producing 1 pup every 2 to 3 years. That's an extremely vulnerable life history, much more vulnerable than an elephant, say. So if they're subject to bycatch overfishing, their populations can be driven down really quickly. So what about these deep sea sharks? Where do they live? So as I said, about half the species of living chondrichthyans live deeper than 200 meters, and we don't know much about most of those. Most of them, pretty much all of the sharks are in the upper 3,000 meters. There are virtually no sharks below 3,000 meters deep in the world's ocean. Of course, the average depth of the world's ocean is deeper than 3,000 meters. But within that top 3,000 meters, the diversity of sharks and their relatives is quite high. So for those of us that are shark nerds that want to study something that's completely new and you never know what you might see, studying the deep sea sharks is like space exploration to us. Now, in terms of the diversity, here's a pretty standard cladogram of the relationships of the living cartilaginous fishes, from the chimeras all the way down to your requiem sharks down here. And so you've got 4 superorders basically, and within the chimeras, the order Holocephalomorpha, these are almost entirely deep sea. There are a few shallow water, but they're cold water temperate species. But these little weirdos, there's only about 55 known species, maybe a few more than that being described now. And they live their whole lives in the deep sea, lay eggs, and we know almost nothing about these chimeras. Now, the next slide I'm going to show, I wish I could hear all your expressions because I guarantee it's going to make you all go, oh, because you're going to see the cutest thing ever on the planet. This was an image sent to me by Christian Jones from NOAA. It was taken from OV, and this is what a hatchling chimera looks like. Ooh, right there. Look at that. Is there anything cuter on the planet than that? I don't think so. That is the cutest animal on the planet right there. That's cuter than a baby panda. What about within the elasmobranchs, so our sharks and rays? We actually have deep water taxa among all 3 of the superorders. So we can't ignore our batoids, of course, because the rays are essentially just pancake sharks. They're proper elasmobranchs like any shark is. And most of the deep water, there are a couple of exceptions, but most of the deep water species of batoids are skates. Lots and lots of skates. Skates are a huge group, nearly 300 species, and most of them are in cold water. Most of them are deep water. For example, these are some of the deep water skates we've caught during our work in the Gulf of Mexico. This one on the bottom right is Rhyella purpura-ventralis, which is a species that the genus Rhyella generally signifies relatively small skates. This is a species that was never known from adult until we started our work in the Gulf of Mexico. It was only known from juvenile right here. And so we're actually in the process of redescribing the species based on the morphology and genetics, including adults, into the description. But there are some other things besides skates that are batoids and are found in the deep sea. This is another clip from a documentary we did with National Geographic. And this crazy looking animal is Plesiobatis davisi. It's the only member of its family, one species. They're Indo-Pacific only, but they're found in the deep sea. And they're related fairly closely to your typical stingrays. You'll see that this thing has a very large stingray spine on it, lives down at around 400 to 700 meters deep in the Indo-Pacific. This is in Hawaii where we're doing this work. You see it's almost translucent. It's got giant venomous spines, which are basically just modified scales. And there is not a single scientific paper ever done on this animal, and they're not hard to catch either. So this just illustrates that this field of deep sea biology with sharks and rays is just wide open. How about this thing? Look at this thing, Hexatrigon. It's the only species of ray that has 6 gill slits. All the others have 5. Isn't that crazy? It's considered a sister group to all of the other stingrays. Again, not a single scientific publication other than if you just look in Google Scholar for this species, what you'll find is that there are just records. First record of the species from this area, second record from this species. Nothing known about its biology or its ecology. The squalomorphs superorder are the dogfishes and the relatives, and this is where the majority of the deep water species reside. Most of those squalomorph sharks are found in the deep sea, and this includes, for example, all of your dogfishes, your gulper sharks, which are these 2 families here. These are some of the species we catch in the Gulf of Mexico, and in fact, about 92% of all the sharks and rays that we catch in our deep sea surveys are squaliform sharks. 80% are just these 4 species right up here, these 4 species of dogfish and gulper sharks. So they really make up the majority. This is a typical one. This is shot from a submarine in Exuma Sound in the Bahamas. And this is Squalus cubensis, pretty typical deep water dogfish, big eyes. They live where there's still enough light to see, so they have large eyes to gather as much of that light as possible, and they're adorable. Of course, in doing this deep sea work, you'll often come across things that are new to science. For example, this is Mariah Flager. Mariah was an undergraduate student in my lab, and then she did her master's degree with Toby Daly Engel. And we described this new species of deep water dogfish from the Gulf of Mexico, which we named Squalus clarkii. The Clarkii name is named after Jeannie Clark, who some of you may know as the shark lady. This is a recent paper that we published with Peter Oster. They stumbled across with an ROV at about 540 meters a dead swordfish, and it was just being consumed by sharks. Turns out the sharks are primarily Squalus clarkii, that newly described species. There is also Serigalius asper right here, but most of them are Squalus clarkii. It's important to remember, though, like I said, most deep sea sharks are relatively small. That means they may not just be predators, they may also be prey. And this also happened during that filming. This was a Squalus clarkii basically feeding on a swordfish, and a wreckfish, a large bony fish, came up and just ate it. Just ate it. So deep sea sharks can be prey as well. What about this guy? I can't talk about deep sea sharks without talking about the rough sharks, the oxynoids. There are only 5 species in this family, and we know almost nothing about them. The only one that occurs in the Western Atlantic is this guy here, and it's only known from a few records from basically ROV and submarine camera footage. We don't know anything about its biology, and it's got a tiny little mouth, and for decades it was assumed that they probably mostly fed on invertebrates, worms and crustaceans and things like that. But then there was a paper that came out in the Mediterranean from one of the rough sharks that suggested that they mostly eat eggs of sharks, of other sharks. This one had little cat shark babies in its embryos in its stomach and bits and pieces of egg cases. And I thought, well, that's just probably just a coincidence. That's probably not their normal feeding mode. And then just a couple of years ago, Britt Finucci published a really cool paper on Oxynotus brunniensis in New Zealand, where she found that actually the most of what was in the stomachs were either egg case pieces or the embryos of chimeras. So this is a deep water shark with a tiny little mouth that's mostly seems to be feeding on egg cases of other sharks in the deep sea. How cool is that? Super cool. Another example of things you just never know that you're going to stumble across would be in 2013, we were doing our standard deep water survey. We were at nearly 6,000 feet deep, very close to where the oil spill occurred. And we caught this thing. This is a giant sleeper shark, Somniosus. It's a Greenland shark. And everybody freaked out. Got a lot of press coverage because everybody's asking, what's a Greenland shark doing in the Gulf of Mexico? Well, if you recall that thermocline I showed you, at that depth, it's 4 degrees Celsius. If you go anywhere around the world at that depth, it's around 4 degrees Celsius. So why wouldn't it be there? But super cool to catch something that's completely unexpected. And that's what really attracts us to study the deep sea. And that just shows where we caught that Greenland shark compared to where the oil spill occurred. Well, and of course, Megalodon still lives in the deep sea. Everybody knows that, right? Yeah, that's not true. Meg went extinct about 2.6 million years ago. And actually, if you go to this little link here, you'll find that there's a video clip where they claim to have caught Megalodon on film, but actually what they're filming is a giant sleeper shark like that Greenland shark that I just pointed out. So Meg's gone, and that doesn't matter anyway because Meg's not all that cool. Another big group, the biggest dominant predator in terms of a shark in the deep sea, everywhere I've done work in temperate and tropical areas, tends to be the blunt-nosed six-gill shark. And blunt-nosed six-gill shark, I would argue, is way, way cooler than Megalodon anyway. And was around, this group of the six-gill sharks were around 150 million years before Meg ever showed up on the planet. And they're still here, and they're super cool. And I'll talk briefly about them right at the end of this coming up. And then there's the last superorder, the gallionomorph sharks, and these include most of the sharks you're more familiar with. And there are a few deep water representatives in every order except for the horn sharks in this group, but it's really dominated by one group in the Carcharrhiniforms, the requiem sharks, and that's this group, the Ciliarhinidae, that are mostly deep water. They're the largest group of, largest couple of families of sharks. They're commonly referred to as cat sharks. Yeah, that joke's getting old. I've used that one a few times. But there are some other really cool deep sea gallionomorph sharks. This is my former student, Mackie Volich, and Mackie did part of her master's work setting deep sea cameras down like this Medusa and this Pegasus in the upper right in the Exuma Sound. And this is one of her little video clips that she took. For some reason it turned green. Apologize, I don't know why. But notice this is real time. This isn't slowed down or anything. This is how slow this animal is moving. And this shark is Pseudotrachus microdon. Before we started doing work in Exuma Sound, there were no records of this. It's one of the false cat sharks. There were no records of the false cat shark in the Bahamas, but we had, Mackie had a few times where they came and actually hung out at her trap, on her camera trap. So really cool deep water species. And of course, there are a few deep water mackerel sharks or lamniform sharks too, things like the goblin shark. And we're finding out, that they're more and more widely distributed all the time. This is one that was caught a few years ago off Key West. And so you think of most of the goblin sharks coming from the West Pacific, Indo-West Pacific, and from Japan. This one was in the Northwest Atlantic. So if you think about the sort of the overall picture of energy flow and everything in the deep ocean, this is an old graph that sort of symbolizes the main patterns, where you basically have this large permanent thermocline where it gets really cold below 1,000 meters. Light also disappears below 1,000 meters, essentially dark. And because of that, biomass tends to decrease as you get deeper. You tend to get two peaks of biomass in that upper 1,000 meters. And that's the result of that upper peak being in the photic zone where photosynthesis is taking place. So you have primary production, fueling it, and then you have sinking particulate carbon that sinks down and provides the nutrients needed for that second peak. Below that, the biomass of animals tends to decrease. Well, in our work in the Gulf of Mexico, we see essentially the same thing. You really see the peak in biomass and species diversity in the sharks, really in about that 300 to 600 meter range is where we tend to see them. So these are the number of sharks we caught per 100 hooks as a function of depth, and you can see they peak at around 500 meters generally. We can divide these deep sea groups of sharks based on their basic ecology or the habitats where they live. Many of the sharks are actually deep benthic. They stay on the bottom, and many of them don't migrate. In fact, in our work, if your hooks are not right on the bottom, within a meter of the bottom, we generally don't catch very many sharks at all. You get more species in what we refer to as the mesopelagic zone or the twilight zone. This is an area where that light is rapidly attenuating, rapidly decreasing. So the sharks tend to have large eyes to allow them to really harvest that really dim light. Most of these don't migrate. And so these are day in the black dot, or nighttime in the black dots, daytime in the white dots for 4 species of deep water sharks in our survey circled in red. Notice there's no real difference between day and night. So they don't migrate vertically. They just stay at the same depth all of the time. And so this includes things like the cat sharks and the dogfishes primarily. Then there are also groups, though, that are deeper than that and that are more in the midnight zone where there's almost no light at all. These tend to have very, very small eyes, and it tends to be dominated by very deep water cat sharks like this one here. This is one of the most common globally distributed cat sharks. They only get about 60 centimeters, 50 centimeters long, very small animals. So they're little dark brown sharks with small eyes. Then we have pelagic migrators. So those that don't live associated with the bottom, but instead migrate in the water column. But their migration may not be as defined as migrating during day and night as you might imagine. These are primarily going to be in the mesopelagic zone because they're mostly following the daily migration of zooplankton so they can feed on them. Most of these are bioluminescent sharks, so sharks that actually produce light, which is way cool in itself. This is the day and night distributions of one lantern shark that we catch in our surveys. You can see during the night or during the day, they tend to be at a very defined depth, don't move around very much, but at night, a lot of variance because they're up and down in the water column constantly as they're trying to feed. So I said they produce bioluminescence. Many organisms in the marine environment produce bioluminescence, but only a couple of families of sharks produce bioluminescence and no batoids. Mostly it's just the lantern sharks and the kitefin sharks that produce bioluminescence. And there are a number of reasons why you would want to produce light. You may think, well, if you're a shark in the middle of the dark black ocean, why would you produce light? Well, one of the main reasons is to obliterate the downwelling light to break up your silhouette. This is a fish and a squid without their photophores turned on or with their photophores turned on. So camouflage using downwelling light. Some work by Edie Witter, who I'll mention in a little bit. And so that's what a lot of these sharks are doing, producing this light to basically break up their silhouette, because most of these bioluminescent sharks are really small species. For example, this is my student, Joanne Emhoff, with one of our main species we catch, Elasmo virens. And this is an adult. Now imagine if you're a little tiny shark this big and you're producing light in the deep sea, you better be camouflaged because you're going to be lunch for something. Of course, you got to be pretty voracious to live out there too. You can see it took this big bait right here. But it's one of the coolest species out there. And it also sort of illustrates another reason why animals have bioluminescence for species recognition. For example, lanternfishes often have very distinctive patterns to their photophores so they can recognize one another, recognize the species. The sharks do this too. They have very distinctive patterns of the photophores on their body. Here you can see in this Elasmo virens, this is an adult male and female. They're only less than 25 centimeters long. They have very distinct patterns of photophores on their side. Now this is, to my knowledge, the only pregnant one that's ever been examined. We caught it in the Gulf of Mexico, and it had 5 term pups inside. I'll zoom in on those. They're only about 9 centimeters long. Imagine that you're in the deep sea swimming around and you see this little 9 centimeter, 3-inch long shark that's got its belly just lit up with photophores. You wouldn't believe your eyes. It would want to be one of the most amazing things you've ever seen. This is one of the coolest things I think we've ever been able to see in all the work we've done in the deep sea. Another purpose of bioluminescence is to create a lure for predation. Of course, everybody knows from Finding Nemo about the anglerfishes that use their bioluminescence to lure potential prey. Well, there's sharks that may do this as well. In '88, Edie Witter published a paper that basically suggested that in the cookie cutter shark, they have photophores all along their belly, but they're missing photophores in one little collar around their neck. The idea is that basically makes them camouflage except for this little collar, which makes it look like a smaller silhouette that could potentially attract larger fishes or cetaceans to that collar with no photophores. And then of course they have these giant teeth, which they then lunge out, and they're essentially parasites, and they take big chunks out of other larger animals. This is a swordfish we caught in the Gulf of Mexico last year. And you can see a very fresh cookie cutter wound taken out of it. And by the way, cookie cutter sharks probably aren't the only ones that feed this way. This is the kitefin shark, the Latius leica. Notice this dentition is very similar, and it probably feeds in a very similar way, which is really cool too. There are many reasons to have bioluminescence, and I'm not going to go through them all, but is to confuse potential predators, either by creating flashes of light to startle a predator, or like this shrimp or a lot of squid will do, create a bioluminescent secretion to basically provide cover for you to escape a potential predator. Now, some of you may have heard about the American pocket shark that was described in a few years ago. This is another shark, obviously way cooler than any white shark or any other thing that you see on a documentary typically. This guy should have its own TV series. So cool. It's called a pocket shark, not because it fits in your pocket, although it could. It's called a pocket shark because it's got a pocket right behind its pectoral fins. And Mark Grace, who described this, sent me some illustrations that he did of how he believes that pocket works. That pocket actually secretes a bioluminescent fluid. And so Mark thinks that they basically create this fluid and it basically cloaks the whole animal in the open ocean, the pelagic zone, and basically hides them from potential predators. How cool is that? Doesn't get a whole lot cooler than that. Then there are also some that migrate along the bottom. So these are ones that basically live along the bottom, but they migrate daily up and down. This includes a lot of things like the gulper sharks here, the six-gill shark as well. Here you can see if you look at their nighttime depth versus their daytime depth, across the board, their nighttime depth is shallower than the daytime. So during the day, they stay deep. Sun sets, they come shallower to get closer to potential prey. And then during the when the sun sets, they go back down into cooler depths to digest their prey. And so this brings me sort of to the last topic that I want to bring up is that I said I'd come back to six-gill sharks because I think they are the coolest species of shark on the planet, to me at least. We've been doing work on the blunt-nosed six-gill shark for a number of years. This is one in Hawaii. And we've basically been, one of the big questions we had from the beginning was, can you even learn about the ecology of these animals without sacrificing them? Can you tag six-gill sharks and release them and they survive and you learn about their biology? Note that dark, big, bright green eye, which is really an adaptation for basically harvesting that dim light. Also notice how flexible this shark is. A lot of sharks are really flexible, but six-gill sharks have essentially become notochordal, which means essentially the vertebral column, it doesn't have really well-defined centra. So it's way more flexible than most other sharks. But so we're curious if you could even start to tag deep sea sharks and get any information, or would they all just die? And so this was the first species that we started working with, this blunt-nosed six-gill shark. And so this is one of the ones we tagged in Hawaii. This is a relatively small one. It's a maturing male. It's about 3.5 meters long. They're nearly neutrally buoyant. They have a very large liver. And so when they're at the surface, if they take in any air, they'll actually float on the surface. So we usually turn them up like a Coke bottle to get the air out, then release them. And they usually swim down just fine. And you can see this one's trailing a satellite tag to allow us to tell whether this animal survived or not. So we've done this a number of places around the world. Some places we can't do it by getting in the water with them like that. This is in the middle of the Gulf of Mexico. This is a huge 5-meter long six-gill sharks, about 17 feet long. As I mentioned, they're among the largest of the predators in the depth range from 200 to 1,000 meters around the world in tropical and temperate environments. And so this one actually, we weren't able to get on board because it was so big. And so we had to tag it by just leaning over the back of the boat. This is about 100 miles off the coast of Tampa, Florida in the Gulf of Mexico at a depth of about 500 meters deep is where we caught this animal. And so we were able to tag it and let it go. Other times we actually bring the animals on board. And so this would be more similar to what a fishery might experience. We did have, there's an issue where a lot of deep water sharks that are caught as bycatch in fisheries, the liver is removed and the rest of the shark sometimes is thrown overboard because the liver is the only thing that's really marketable for its oil. The rationalization has often been they wouldn't survive anyway, so they're just taking what they could. We have been referring to that as livering. And so we wanted to see if we bring these animals on board like they would on a fishing boat, do they actually survive? Could they be released and survive this? So we've tagged 20 something of these in 5 different places around the world: the Gulf of Mexico, Bahamas, off Virginia, Hawaii, Ascension Islands. Across the board, one of the things that we see, these are 2 animals from the Gulf of Mexico, is that they basically come shallow at night and they go deeper during the day. And that is very regimented, and it's everywhere we've looked at them. The black here is nighttime depths, the red is daytime depths. And so they basically dive down as soon as the sun comes up and come up the slope as soon as the sun sets. Almost all of ours that we tagged survived, which was a surprising result. The only ones that didn't survive actually got eaten by other sharks almost immediately. And so you may wonder, how do I know it got eaten? So here is an example of a 6-gill shark that survived on the left and one that got eaten on the right. You see this one, if I zoom in just on one 24 or 48-hour period, if you draw a line of when this animal met its minimum depth and its minimum temperature, so of course temperature is going to follow depth, you can see there's almost no lag because the temperature, the thermometer basically measures exactly the temperature within a few seconds. And so there's no lag. But in this one that got eaten, there's actually a 6 to 8 hour lag, meaning that this tag is inside the belly of a big shark. And because of the thermal inertia inside the belly of that shark, it takes 6 to 8 hours for the internal temperature to cool down after it dives to the bottom. Now, there are reasons why it warms up faster that I don't have time to get into now. But basically, this is an animal that got consumed. But by and large, they all survived, and they also showed the same sort of pattern of going deep during the day, shallow at night, although the depths actually changed from region to region. But if you plot the temperatures associated with those depths, so these are actual temperatures measured by the tags at different depths in these different regions, what you see is that these dives are all to the same depths. No matter where they are around the world, they're coming to about 17 degrees during the night, and they're down about 5 degrees during the day. And so that temperature, the depth of the thermocline, determines how deep they're going. So in the Bahamas, Exuma Sound has a very depressed thermocline, so that's why they're going a lot deeper there than they are up, say, in the Hawaii or in the Gulf of Mexico. One of the other things we noticed is that across the board, there seemed to be about a 2-day period after we released the animals where they behaved what we perceived as being abnormal. In other words, the depth use pattern the first 2 days was different than it was the rest of the tracks. And so we assume this is a recovery period, so that we assume there is trauma associated with being brought to shallower depths. Pressure changes, even though they don't have a swim bladder, there are still some physiological issues with the pressure changes. Temperature changes because we're bringing them up into temperatures that are way warmer than they normally experience. Physical trauma from being brought on a boat, for example, although what we found is if we tag them and release them, just leaving them in the water, or if we brought them on the deck of the ship, it made no difference in their survival. But we were curious if this was representing a true recovery period. And so as a control, the way you would test that is to, of course, not catch them. Don't bring them to the surface. Go to where they are and try to tag them at depth. And it's something I've wanted to do for about 10 years, and sort of finally got the opportunity the last couple of years working in collaboration with Ocean X and the Cape Luther Institute to try. We convinced them to let us basically strap spear guns to the front of a submarine with satellite tags attached to them and to try to tag sharks at depth. Also, it was the first time I've ever observed sharks in their natural environments. It's really cool. I was very privileged that my very first dive in a sub ever was with Edie Witter, who's right here. Edie was the author of that cookie cutter shark paper I just showed you, and she's the one who made the bioluminescent lures that the giant squid came after, and she's probably spent more time in research submarines than any human. And so great privilege for me to be able to do my first dive with her. And so we went to the bottom of the ocean. We're sitting at about 550 meters deep after sunset, because remember, these sharks are coming up from about 800 to 1,000 meters during the day, up to 400 or 500 at night. So we want to be able to intercept them because the daytime depths are too deep for the submarine. So we're just sitting in the dark. If your screen's black, it's supposed to be. We're sitting in the dark and just waiting and waiting. And we actually have a little low resolution camera that we can actually barely see through a red light what's happening. We turned the lights off because we didn't want it to change the behavior of the animals. Well, at one point, that little low resolution, low light camera went completely black, and we didn't know what was going on. And Edie quickly said, "Turn the lights on of the sub." And when she did, this is the first thing we saw, was this giant 6-gill shark basically trying to eat our spear gun. This animal had basically, the reason the screen went black on our low light camera was that its body was pressed against right up against the screen, right up against the lens, and so it was blocking it. And so this is the first time I've ever seen a 6-gill shark in its natural environment. This is a GoPro that Brendan Tolwer strapped to the outside of the submarine. This is the view from inside the submarine. Edie actually shot this video on her cell phone, I think. You can see after it came up from the bottom of the sub, it swam over our head. It'll slowly come around and pass right in front. This is a large male. We also saw a large female on this trip. Just a great experience to be able to actually see a 6-gill shark in its natural environment. We actually had lasers attached to the spear guns so that we could sort of aim, because there is a shot zone where I want to tag the animal without harming it. This animal passed just a little bit too low. You can barely see the lasers on its side. It's a little too low to take a shot, so I didn't try to tag that one. We had several attempts over 3 cruises and a lot of trial and error. But this is from the very last trip that we did, and my colleague Gavin Naylor was in the sub during this time. And this is an enormous 6-gill shark. And you can see it came up. It's probably 5.5 meters long, maybe bigger. It would probably be one of the largest ever measured if we were able to get an accurate measurement on it. And it was just basically pushing the sub around. And we had that happen several times, just giant, impressive animals. And during this dive, Gavin actually was able to tag one 6-gill shark. So we have actually proven the concept that we can tag these sharks at depth. Here you can see it's going to come right into the controls. That's a little nerve-wracking when you've got this giant head under your feet, pushing the sub around, looking around at you, looking inside. But overall, an incredible experience to sit on the bottom and watch this animal just swimming around naturally. And so we're hoping to do more of this with other species and actually develop better methods for actually tagging these animals at depth to figure out what they're doing naturally without having to catch them. That would be ideal if we could do that. If you want to see the whole, there's about a 12-minute documentary that OceanX put together with National Geographic. You can go to this link here and watch it. And that's all I'm going to talk about. If you want to see any of our other work on deep sea, my favorite documentaries I've ever done have always been with public television. This one on Changing Seas, Creatures of the Deep, you can watch for free online. You can also watch this Dispatches from the Gulf episode from the Secrets from the Deep, this one's called. And it's part of the Journey to Planet Earth series. It's been narrated by Matt Damon. You can watch that for free as well. They did a really good job. The last thing I'll mention is another shameless plug for the shark book that we just had come out. If you're interested in shark biology, feel free to pick up a copy. It's available now. And with that, another thank you to Elasmo Week for inviting me today and for the introduction from David. And thank you all for listening. And I hope you have a great weekend. And I hope to join you Sunday for the question and answer session as well. And that's it.
Great. Thanks, Dean, for that really interesting talk. Just as a reminder for everyone else, we have another talk coming up right now on the Miss Elasmo channel. And as Dean mentioned, Sunday evening, we're going to be having a live Q&A session with all of the Elasmo Week speakers. So if you have a question for Dean about anything that you saw here, you can comment on the YouTube video you're watching or tweet it at Elasmo Week. Despite me trying for 9 years now, Dean does not have his own Twitter yet. But if you can tweet at Elasmo Week or leave it as a comment on the YouTube video,